we are going to discuss about the second plant growth regulator, which is gibberellin. Okay, so first we discuss about auxins, so now we are going to discuss about gibberellins. Okay. So here gibberellins are, there are 100 different kinds of gibberellins found from many organisms like fungi and other plants and etc. So it is named as GA1, GA2, GA3 and so on. So the main gibberellic acid, the main gibberellin is gibberellic acid, okay, which is GA3. Okay. So this gibberellic acid was first discovered and it was widely studied. Okay. So these gibberellins increase the length of the axis. It is generally used to increase the length of fruits, stems and increase the internodes and everything. So it increases the length of grapes stalk. So we get long grapes due to the application of these gibberellins. And these gibberellins cause apple to elongate and improve its shape. Okay. So these gibberellins also delay senescence. Senescence is nothing but the older it becomes old, the plant parts becomes old, which we have already seen. So therefore the fruit can be, it delays senescence. Okay. So therefore it delays the plant to become old. Okay. So it will be retaining in the maturity level itself. So therefore the fruit can be left on the tree for a longer period. Okay, longer market period or anything. So after that this gibberellins speed up malting process in brewing industry. In brewing industry it speeds up the malting process. Okay. And sugarcane, when these sugar canes with the application of gibberellin increases the length of the stem. Okay. These sugar cane increase in heights and everything. So therefore 20 tons per acre is increased due to the application of gibberellins in sugar cane fields. Okay. And it fastens the maturity period of some seeds. For example, if you take the juvenile conifers, the maturity period of that conifers will be fastened by the application of these gibberellins. Okay. So therefore early seed production will happen and therefore it is useful for useful replanting. And these gibberellins also promote bolting. Bolting is nothing but internode elongation before the period of producing buds. Okay. So the internode elongates before the production of buds. So that is known as boltings. So it is found in beet and cabbages. It is widely applied in beet and cabbages. Okay. So next we will see the next plant growth regulator, which is cytokines. So here these cytokines have effect on cytokinesis. Okay. We have already seen the division of cytoplasm, so these help in that. And it was discovered as kinetin. So these cytokines were discovered as kinetin. Okay. So these kinetin does not occur naturally in plants. Okay, it does not occur naturally. And the establishment of kinetin led to the discovery of zeatin. So this zeatin was found in corn kernels and coconut milk. Okay, it was found in coconut milk and corn kernels and etc. So this zeatin also has same functions whatever the kinetin has. Okay. So natural cytokinins discovered from some natural cytokinins were also discovered. It was discovered from rapid cell division areas like root apices, developing shoot buds and young fruits and etc. Okay. So what does these cytokines do? These helps to produce new leaves, chloroplast in leaves, and it helps to produce lateral shoot growth. Okay, it produces lateral shoot growth and adventitious shoot formation and everything. So it helps in the production of these all parts in plants. Okay, and it also helps overcome the apical dominance. Apical dominance, which you have already seen that auxins. So apical dominance is nothing but the dominance of apex, the apical dominance which leads to the which does not lead to the formation of the lateral shoot, lateral buds. So therefore we have to cut the apex. So therefore the lateral grows in the, for example in tea plants hedge making is used. So like that, it helps to overcome the apical dominance. So apical dominance is overcome by the application of cytokines, okay. And it promotes nutrient mobilization. So here nutrients are mobile, very mobile, it is actively mobile, which helps in the delay of senescence. So therefore, the older parts do not die soon because of the rapidly mobile elements. It induces rapid mobility of elements. So therefore, always the leaves and everything which are still mature, they will be still mature because of the mobilization of elements. So when there is no elements, the leaf gets wilt and falls. So when there is mobility of elements, the leaf do not fall and senescence is delayed. Okay. So next we will see the next plant growth regulator, which is ethylene. Okay. Ethylene is a gaseous plant growth regulator. It is produced by tissues undergoing senescence and ripening fruits. Okay, there are those which are fruits which are ripened, and those tissues which are undergoing senescence. Senescence is nothing but the falling of stage, the older stage. So these tissues and ripening fruits produce ethylene in them. Okay. And it also enhances the horizontal growth of seedlings. Seedlings, swelling of axis, and the axis swells, and apical hook formation in dicot seedlings. So in dicot seedlings there is apical hook. In dicot seedlings there is an apical hook formation which occur. Okay. And it promotes senescence. Senescence is nothing but it promotes the holding up the older process of the plants. And abscission of plant organs. Abscission is nothing but the weathering of plant organs. So this one promotes. Whereas the cytokines and gibberellins, they delay senescence, but here it is promoting senescence. Okay. Abscission of plant organs like leaves and flowers and everything. So it is highly effective in fruit ripening. So this ethylene is applicated for the fruit ripening of various fruits like bananas and everything. It enhances the respiration rate in ripening fruits. Okay, so in ripening fruits, this ethylene enhances the respiration rate of these fruits. Okay. So this enhancement of respiration rate is known as respiratory climactic. Okay. And this ethylene breaks seed and bud dormancy. It breaks dormancy. Dormancy is nothing but the inactive stage of the seed.
Okay.
Okay. So factors which do not influence the germination of seed is known as it is at a dormancy stage. So dormancy stage is overcome by the application of water, sunlight and everything to the seed. Then the dormancy condition is overcome. So here it breaks seed and bud dormancy, initiates germination in peanut seeds, sprouting of potato tubers and etc. in the application of ethylene. Okay. And it promotes a rapid internode or petiole elongation. Okay. So petiole or internode, it elongates by the application of ethylene in deep water rice plants. Okay. So the upper part of the shoot or the leaves in aquatic plants, by the application of ethylene, the upper part of the leaf or shoot, they remain above the water. Okay, they just remain above the water. So we have discussed some characteristics of ethylene. So next we'll discuss more about ethylene, more characters about ethylene. Okay. We are going to discuss about ethylene.
Okay.
Our fourth plant growth hormone. So here it promotes root growth and root hair formation in plants. Okay. So this ethylene is a gaseous hormone. It helps to increase the observing surface. Okay. It initiates flowering. It initiates flowering in mango plants and etc. Okay. So by the formation of root and root growth and root hair formation, these help increase the absorbing surface area and okay, by the application of ethylene. Okay. So this ethylene is the most widely used plant growth regulator in agriculture because these have so many functions. So these ethylene are widely used in agriculture. And this ethylene is a white compound source of the white compound which is source of ethylene is ethifon. Okay, so this one is an aqueous solution of ethylene. So it is widely absorbed and transported within the plants and releases ethylene slowly. Okay, so this ethifon, it is widely absorbed and transported by the plants, and this ethifon after going into the plants, it releases ethylene slowly. Okay. So this ethifon it fastens the fruit ripening in tomatoes and apples. So it ripens faster if the application of ethifon is given to the plants. Okay. And accelerates abscission of flowers and fruit. So this one promotes senescence. So because it accelerates means it fastens the abscission of flowers and fruits. Okay, abscission is nothing but the weathering off of flowers and fruits. And this ethylene promotes female flowers and cucumbers. Okay, it promotes only female flowers and cucumbers and increasing the yield of cucumbers. Okay, therefore increases the yield of cucumbers. So this is about ethylene, and next we will go with the fifth plant growth regulator, which is abscisic acid. Okay. So now we are going to see about the fifth plant growth regulator, which is abscisic acid. So this regulates abscission and dormancy in plants. So abscission is nothing but the weathering off, and dormancy is nothing but the dormancy of seeds. And under unfavorable conditions, the seeds undergo a stage called dormancy, and that is what it is. So it regulates the dormancy period. Okay, it regulates to withstand unfavorable conditions of seeds and everything. So it is abscisic acid is a plant growth inhibitor, which we have already seen in the last session. So abscisic acid inhibits seed germination. It inhibits seed germination. It does not allow seed to germinate. And it stimulates the closure of stomata. It stimulates the closing of stomata in the epidermis and increases the tolerance to various kinds of stresses. Okay. So therefore it is known as stress hormone. And it acts as antagonist to gibberellic acids. Antagonists are nothing but it acts opposite to gibberellic acids. Okay. So next we go to the fifth topic, which is photoperiodism. The fifth topic, which is photoperiodism. So this photoperiodism is nothing but some plants. Okay, some plants have periodic exposure to light. Some plants on exposure to light, they induce flowering. Okay. So some plants measure the duration of light, or how long the light is fall on these plants. So some plants can measure these durations. And exposure to light more than a well-defined critical level, critical duration. If it is more than the critical duration, it is known as long day plants. And exposure to light less than critical duration is known as short day plants. Okay. And in some plants there is no relation between critical duration and light conditions. Okay. So those plants are known as critical duration and light conditions, nothing but no relation between the exposure to light and the flowering. So those plants are known as day neutral plants. Okay. So after that, these are all based on light conditions. So after that, they prove that dark period also play a major role in plants. Okay, dark period also gives equal importance in these plants. So this condition of plants to exposure of so the plants which regulate their flowering with response to light and dark conditions is known as photoperiodism. Okay. So this photoperiodism is nothing but it has to measure, or it has to absorb that exposure of light, right? So that site of perception, site of absorption of light, of dark and light duration, you should know it is dark visible light. So that site of perception is present in leaves. Okay. So leaves have some hormonal substance. It migrates from leaves to the shoot for leaves flowering with respect to this light and dark conditions. Okay. So for example, if you see that radish, it is a short day plant. Okay, it needs critical duration below. Okay, so it is below the critical duration. Light exposure is below the critical duration. Whereas in Spanish, henbane, sugarcane, wheat, everything are long day plants. they need light more than critical duration to flower. Okay. So now we have seen photoperiodism. So next we will see the last topic, which is sixth one, vernalization. Okay. So now we are going to see the last topic, which is vernalization. So vernalization is nothing but some plants, they flower with respect to the temperature. Okay, on exposure to low temperature, they initiate flowering. Okay, they flower enormously. So this condition to flower at low temperature is known as vernalization. Okay. So there are two varieties, winter varieties and spring varieties. So winter variety plants flower only in winter. Spring variety plants flower only in spring. So if you take winter variety plants and plant it in spring varieties, it does not flower because the condition of low temperature needed here. Okay.
It's like taking the camel from the desert and bringing it to
tropical regions. So and there are some other plants called biennial plants. Okay. So these normally flower and die in the second season, biennial plants. Okay. So so the year two times they can harvest. Okay, these biennial plants. So normally they flower and die in the second season, which is also known as monocardic plants. So examples are wheat, cabbages, and carrot. So if these plants were given a low temperature conditions, they can retain for long period of time and they flower for long period of time. So that is the use of this vernalization. Okay. So with this the chapter completes. Plant growth.
Low temperature estimated value can be used.
No, not estimate. Different plants have different values.
Wheat have other cabbages and carrot.
So with this the plant growth and development chapter ends. Okay. Thank you. If you like this video, please subscribe and click the bell icon which is nearby. And if you have any doubt, please leave them in the comment section.
Thank you. Kritika and I signing off.